For those of you who might not be familiar with our series, we meet every Monday at this time here, and our schedule can be found online at www.bec.ucla.edu. A brief preview of next week's talk. We're going to have David Liu from the UC San Diego Department of Psychology, and the title of David's talk is Asking, quote, do X have a theory of mind? Is not precisely the right question. Mental state understanding is not yes or no. So that's going to potentially be a very interesting talk. And today, it is my great pleasure to introduce John Novembre from the UCLA Department of Ecology and Evolutionary Biology and the IDP in Bioinformatics. And I should mention that this talk has been generously co-sponsored by the UCLA Center for Society and Genetics. So without further ado, John.
So I'm going to begin with a geography trivia question. These two locations, Shrewsbury, England, and Murray's Publishing House in London, does anybody know how they're connected? Darwin, right? So Darwin was born in Shrewsbury, England in 1809, and he published The Origin of Species at Murray's Publishing House in London. And between 1809 and 1859, some very important things had to happen. It wasn't just that, even though these two events took place in a small area of the world, that it's not only these two locations that are relevant in making the origin of the species possible. What's very important was that Darwin made a giant voyage in between, traveling between these two locations, the voyage of the Beagle, which we're all, of course, very familiar with. And the important point about this is that he got to, by traveling, see the geographic distribution of many forms of biodiversity. And that gave him a tremendous amount of insight about evolutionary processes and indeed helped him form the theory of natural selection. So this is just to reiterate the point that much of what is crucial in evolutionary biology, or a great source of insight, is the geographic distribution of biodiversity. It's been a key source of insights, not just for Darwin, but for generations of biologists, and it's just as true today. And much of what my research focuses on is on developing methods for studying the geographic distribution of genetic diversity and trying to understand what we can learn from those patterns. So today I want to talk about two different areas that involve studying the geographic distribution of genetic variation. So one is looking at large single nucleotide polymorphism studies in geographically dense samples. I'm going to show how that's allowing us to understand population structure at very fine spatial scales. And in particular, I'm going to talk about work I've done on the population structure of Europeans. And then second, I want to talk more about evolutionary processes in humans and how population structure plays an important role in determining how humans respond to novel selective pressures and, in turn, help determine the genetic basis of adaptive phenotypes. And to do that, I'm going to talk a little bit about lactase persistence, skin pigmentation, and spatial patterns that have been observed in the human genome diversity panel. So first, I'm going to start by talking about Europe. So the main question that many human population geneticists have been asking for some time is what can patterns of genetic variation tell us about the demographic history of European populations? This is a schematic from a review by Guido Barbigiani and David Goldstein from 2004 that shows multiple large-scale events that are thought to influence the genetics of European populations. So first is the initial colonization of Europe 40,000 years ago, represented in these red arrows. This is the original out-of-Africa movement into Europe. Then subsequently, there were, during the Pleistocene, glacial expansions and contractions that's represented by these purple and human population movements that tracked these expansions and contractions, and that were thought that would have taken place around 18,000 years ago, the last glacial maximum. And this would have pushed human populations into refugia, represented here by these purple circles. And then after the expansion of their retreat of the ice sheets, you would have had humans expanding northwards. And then the next major event is thought to be the Neolithic expansion, the expansion of agriculturalists from the Mideast and Near East into Europe 10,000 years ago, potentially replacing the former inhabitants, or either completely or partially, or not at all. It's a highly debated aspect of human population history, and particularly genetic evidence for this Neolithic expansion. So I'll be coming back to that later. Okay, so the Europe is one of the best studied populations in humans in terms of the sampling. It's partially because it's been very easy to obtain samples for a long time from Europe. In fact, it might be one of the most well-studied populations in any species in terms of the population genetic patterns. So in some ways, I sometimes like to think of Europe as a model system for demographic inference. We have so much data from these populations that if we can't make progress in understanding the history of these populations, it makes it raises questions about how well you can do in other populations as well. So, and an important part of this is not just for understanding human history, to admit that a lot of my interest in this and a lot of the applications for this kind of inference and understanding is for the fact that it provides essential background for a few different applied areas. One is understanding potential signatures of natural selection. So a key issue in evolutionary genetics is that to understand patterns of natural selection, you have to understand the background demographic processes, what the patterns of genetic variation look like due to genetic drift, migration, population history, before you can detect what are the outlier signals that are due to natural selection. Also, in genome-wide association mapping, which is a big effort within the medical genetics community to understand the genetic basis of disease traits, it's very important to understand what the population structure of Europe looks like. And then also we have this very recent growth of many commercial enterprises that aim at inferring customers' personal ancestry, saying where an individual comes from after they submit a DNA sample. And so how well some of these methods can do depends on patterns of population structure. Okay. Oh, and I skipped over. I meant to add in. This is yet another kind of layer of the of migrations on top of those three major events that I showed before of the initial expansion into Europe, the glacial movements northwards and southwards, and then the Neolithic expansion. You also have in historical times large numbers of migrations that might be influencing the genetic patterns within Europe. So the results that I'm going to present are the product of analyzing a very large data set that was organized by Matt Nelson from GlaxoSmithKline. And the data that was analyzed is in collaboration with Carlos Bustamante's group at Cornell and Sven Bergman's group at Lausanne. So the data consists of 1,400 European descendant individuals with data on the country of origin for at best all four of their grandparents. And what I'm going to present are results where the individuals are labeled with an ancestry label. This ancestry label is assigned according to the origins of the grandparents. And if the observed grandparents had more than one origin, what we did was just not put a label on that individual. So we're essentially excluding that individual from the plots that you'll see. Okay. Because if we had observed grandparents from more than one origin, it's difficult to sort of summarize where they came from, so we just left that off. And in one way you can think about this is looking at the population structure of grandparents that come uniquely from one location. And so after assigning these labels and looking at how many distinct labels there were, we had the individuals represent 37 unique populations. All right, they're not necessarily populations in a closed biological unit sense of the word, but just meaning distinct labels determined by where the grandparents were reported as coming from. So this analysis is based on 197,000 SNP loci, and so I need to explain what a SNP locus is. A SNP locus is a single nucleotide polymorphism, and so if we align 4 different chromosomes taken from maybe 2 human individuals, that would, because we're diploid, we'd have 4 chromosomes, and we would see that many of the locations, in fact most of the locations in the human genome are actually absolutely uniform across individuals, and some of them vary. and they vary with just a single nucleotide difference, and that's called a single nucleotide polymorphism. They typically have two alleles, so here a C and a T, here a G and an A. And probes can be designed to interrogate what allele is carried by each chromosome. And these probes can be layered onto these silicon-based chips. And what's really impressive about these chips is that a very high density of probes can be layered onto the chips, so that you can get around, commonly now, 1 million SNPs on each of these chips. And that's sort of revolutionizing human population genetics right now. And the costs are low enough that it's becoming routine to do thousands of individuals in these surveys. So what you end up with are data where you have, for each individual here, is represented by a row. Each column represents a SNP, and if we represent the 3 possible genotypes of that SNP by a 0, 1, or 2, we get this matrix of genotypes. And the really amazing thing about it is that we have this matrix has thousands of individuals and hundreds of thousands, and even in the most recent chip designs, millions of columns. So this is very rich data, but it's hard to know what to do with it. It's challenged a lot of the the generation of statistic modeling had been developed for smaller data sets. Essentially can't handle this large of a data. And so many people have been turning towards very simplified multivariate statistical methods, such as principal components analysis, to understand this data. So really briefly, what principal components analysis does is that it takes this large matrix, which again for each individual we have hundreds of thousands of observations, and squeezes the columns together. We're going to actually do a linear combination of the columns that will result in a single number for each individual. And this single number is going to be such that it describes a lot of the variation that exists in this matrix. So there's a sort of an optimization problem that PCA solves. It decides amongst all the linear combinations of the columns, what is the linear combination that results in the most variation in this first number. And then it does that iteratively to capture the remaining variation that's not captured by this first number, so we can get a second set of numbers. And we can keep doing this actually up to, for many, many different PCs, and get higher dimensional representations of this matrix. Okay, so you can also think of it as a form of data compression, because we're compressing all this data down to 2 numbers. And it's been shown to work pretty well in many cases. So here's an example where I actually ran PCA on this data set, and I get 3 distinct clusters. And that's actually quite nice, because in this data set I actually simulated it. So I know, I decided, I made a miniature world where I know what the answer is. And for this data set, I generated it under conditions where I had individuals from 2 populations and a set of individuals who were admixed between them, meaning when I say admixed, that they had ancestry from the 2 different populations. So it makes sense that we have the 2 different populations represented and the admixed individuals appearing somewhere in between those 2 source populations. Okay, so before I show the results of applying PCA to that European data set that I described, I want to describe a little bit more about that data set, just so I can be totally clear about what these individuals, which individuals actually, what kind of biases might exist in the data. So we started out with 3,192 individuals that were given genotyped, and then they had these were individuals of European descent was how they came to us. And then they had labels on them about where their self-reported origin was, and many of these were outside of Europe. And so those were excluded. Then individuals with mixed grandparental ancestry, this is what I was describing before, that reduces down another 500 individuals. Then there were, if you look in the data, you can find individuals that appear to be closely related to each other. The goal of the study was to sample unrelated individuals, but in some cases you get cousins who enter the study. And so this step excludes those, and that's important because if you have very closely related individuals because they're cousins or brothers or closer, or not parent offspring, it can greatly bias the patterns that you see in these PCA plots. So let's see, so then we exclude people with relateds. Then this was done early on. There was a preliminary PCA run where individuals that were seen to be extreme outliers were removed. And this was actually done on a data set that included more than the Europeans. And the thought is that most of these were due to genotyping artifacts. So the chips don't always work, and so sometimes you get very aberrant patterns, and so where the individual shows complete outliers, and those are dropped. And then finally, the data set had a very large number of Swiss-French individuals and United Kingdom individuals. And that is because two of the main sampling centers were in Lausanne and in London. So because PCA is also sensitive to uneven sampling across populations, we thinned those down. Okay, so let's get to actual results, which is if we plot each individual in PC1 versus PC2 space, and we give them these two-letter labels to represent each individual. So this is ES is an individual from Spain, IT is an individual from Italy, FR from France. Okay, we can see this general plot of the individuals. And what I've done to help make the patterns more obvious is to plot the median position of each group of individuals with a large dot. So here this group of French individuals is summarized by this large dot that says FR. And if you have a good sense of geography, you'll notice that this, of course, looks a lot like a map of Europe, where we have Spain and Portugal, France, Switzerland, Belgium, these are British Isles and Ireland, Germany, Austria, and so on, going eastward. And so this was really stunning to us and to many other human population geneticists, because it was thought that at this sort of spatial resolution that human populations are too well mixed to be able to distinguish any relationships between populations. And human populations still are very well mixed, or even well mixed at a global scale, but the very high number of markers that you see here allows you to see even this very subtle population structure. So population geneticists like to measure population structure with a number called FST that varies between 0 and 1. 1 being the maximum difference between populations that's possible, 0 being what's called a panmictic population, meaning that there's no subpopulations at all. And for European regions, if we group these into broad regions, the difference is 0.004. So that's very, very low value of FST. Another way to look at this for people familiar with PCA is that you can ask what proportion of the variation in the total data set is explained by layering individuals on these two axes. PC1 proportion of variation explained is 0.003. PC2 is 0.0015. Okay, these are very small numbers. So that reflects that there's a lot of variation going on in this data set that has nothing to do with geography, but the main patterns that are in it that differentiate the individuals and relate them to each other is geography. And now let's look at, there's a couple interesting outliers. So you might notice this SK here. This SK is for Slovakia. Okay, so Slovakia should be sort of up here with Austria and Hungary, but it appears to be down here near Italy. And that might make you wonder about, is there some interesting population history that connects Slovakia and Italy that we didn't know of before this study? But what's really going on here is a sample size issue. We only had one individual who's Slovakian, so the median position of one individual is just the location of that individual. Okay, so this individual was likely either mislabeled as Slovakian, was actually Italian, or maybe it was a Slovakian individual where we didn't have the grandparental information and had Italian ancestry. So it's, we can't draw much from that. These are some of the other populations that have low sample sizes, but you can see some of them work quite well. Like this is Denmark, actually sits where it should. We had just one Finnish individual, which is interesting, placed here. This is interesting relative to other studies of Finnish groups where the Finns appear as very dramatic outliers. Again, we have just such a small sample size that for the positions of these locations, we can't be too sure. And then separately, we have a few linguistic isolates that are interesting to think about. So, for instance, Hungarians speak a Finno-Ugric language, which is not Indo-European, and so on the basis of language, you might have thought that Hungarians would sit outside of the most of the Indo-European populations that are shown here. But you can see that they nicely do sit near Austria and the Czech Republic and Serbian populations, so that they are close to where they should be geographically, which is interesting from perspectives of thinking about the coevolution of linguistics and genetics. Yeah.
Could you define again PC1 and PC2? How you extract these from the data?
Okay, so so one way to think of it, the way I summarized very quickly was to think of it as a linear combination of all of the columns of your of that data matrix, such that the first variable explains the most variation of all possible linear combinations, and then you look at the residual variation after looking at PC1 and pick a second linear combination that explains the most, has the highest variation, highest variance possible across the column. Another way to think about it, though, is if you think about the calculating a covariance matrix between your individuals. So you have a large matrix that's individuals by individuals, and you take the eigenvectors of that matrix. The eigenvectors of that matrix are related to the PC1 and PC2 scores for each individual. So the way to think about it is as a summary statistic of the covariance matrix between individuals. Does that help?
Yeah.
Okay. Okay, any other questions for it? Okay, so so again, this is interesting from these oh yeah, so we got Hungarians located there. An interesting one is Romanians appear slightly towards the west of where we thought they should be originally, and if you look on the map here, Romania is sort of to the east of a group of former Yugoslavic populations, and including we have individuals that the best label they have is Yugoslavia. So, and what you see here is that it appears to the west. We thought, oh, this is interesting because Romanians speak a Romance language, and we thought this might reflect the influence of their linguistic connections to Romance speaking populations. But actually, if you do a bootstrap approach to assess the significance of this, it actually turns out not to be significant. There's enough jitter in these points relative to where they are on the map that this flipping of Romania over to this side is not actually significant. Okay, but we are able to detect the subtle differentiations. So here you can see within Switzerland, if we break down Swiss individuals into French-speaking, German-speaking, and Italian-speaking, we can see that they have a subtle levels of differentiation such that the French-speaking Swiss look more like French individuals, the German-speaking Swiss look more like German individuals, Italian-speaking Swiss look more like Italian individuals. And this is just really striking. You can get this resolution from genetic data, and it's now leading to many other studies that are seeing similar patterns. So this is within Finland, a very dense sample that was recently analyzed by Chiara Sabati and actually colleagues here at UCLA. And this is large regions of Finland are colored here and represented again in this MDS plot. MDS is multiple dimensional scaling and is very similar to principal components analysis. And you see the same kind of geographic relationships or similarities between the summary of the genetic data and the geography. Here's yet another example where This is Iceland, and we have populations around the ring of Iceland. There's apparently hardly anyone that lives in the center of Iceland, and so the same geographic arrangement of the populations around this ring can be seen in this PCA plot. And here, though, I have to make note that they make a very stringent cutoff of how they label the individuals. So with the European study, we talked about excluding individuals who had evidence for mixed grandparental ancestry. Okay, and so that's in some ways like looking at lineages that have been in each of these populations for 2 generations, right? It's like looking at the origins of the grandparents. Here, the Icelandic population is very well studied in terms of pedigrees, so they get this result when they look at individuals who have at least 16 of their 32 fifth-generation ancestors from a particular region. Okay, so not very recent, and but still striking that you have this very fine-scale population for whom was a very small area, right? I want to ask, why do these PCA plots resemble geographic maps? And this is something I've worked on. A lot of what I do is focused on methodology and how do we study genetic variation. And so I actually got involved first in this research by trying to understand PCA maps. So I want to introduce you to those. This is a different way of looking at the results of a PCA plot. So if we take all the individuals from a certain geographic location and color the country that they're from by the median of their PCA value, we can then get these maps of the PC values. So now we've gone from an individual-based analysis to one that groups individuals and plots their median values on these maps. And what we see for PC1 is this northwest-southeast gradient of the PC1 values. What we see for PC2 is this southwest-northeast gradient. And what this means then is that if we plot individuals on PC1 and PC2, this is kind of acting like like an x-axis and a y-axis. It's going to just replicate the geography of Europe. So that's just a very proximal explanation of what's going on, but I bring it up because it's going to help us understand a little bit deeper what's going on, or at least some of the mathematics, which is that if you simulate PC maps from Steppingstone simulations, Steppingstone models are models that are used in population genetics when describing a lattice or a grid of populations. So imagine, they're called stepping stones because the model was invented by Muto Kimura, who was a Japanese population geneticist and thought about stepping stones in Japanese gardens. But imagine a bunch of populations arrayed in a grid, and they're each exchanging migrants from one population to the next. Okay, it's a very simple model of populations, of spatial population structure. And what I did was I simulated a large number of these individuals from one of these grids, and then calculated the PC1, PC2, PC3, and PC4 maps for one replicate of the simulation, then for another, and so on, for 10 of these. And you see a common feature of these simulations is that you get these gradient patterns in PC1 and PC2, and the direction in PC1 changes due to sampling variation. It's the same exact simulation, but we're just running it again, and we see the direction changing. But a constant part of this is that PC2 is always perpendicular to PC1. Okay, and so it's a very general feature of these Steppingstone models, but it's in fact more general than that. You can actually look at this more mathematically and show that as long as if you have a lattice model with uniformly spaced populations and the covariance function decays exponentially with distance, then the sample covariance matrices, those individual by individual matrices that I was talking about, have very specific mathematical forms called toplet structures or block toplets with toplets block structures for two-dimensional habitats. These special mathematical matrices end up having special eigenvectors, and those eigenvectors, when plotted on maps, have these gradient shapes. or if you plot the individuals, they reproduce maps. I'm gonna, usually my talk spends a lot of time here, but I'm not gonna do that. So the important thing to realize is that this is kind of common to spatial data, is that you'll have these kind of gradient patterns. And you can get distortions from them if you have uneven sampling or if you have more complicated dispersal. So anisotropic dispersal, where the dispersal means that the dispersal is not the same in each direction, then you can get distortions of these. Really what I'm trying to make the point about is that these patterns arise very generally from when you have cases where genetic similarity decays with distance. And so this is a representation of that from the European data. This is a genetic correlation, which basically is a measure of genetic similarity versus geographic distance of individuals in the populations. And you can see here how it decays. And so really what's going on here is that given 2 dimensions, the best way for PCA to represent that you have spatially structured data is to arrange all the individuals geographically. Okay. Now this might seem like I'm going into a lot of detail on just one result, but one of the reasons it's really interesting to get at what's going on with PCA is that it's had a long legacy in population genetics. And in particular, some of you might recognize some of these classic maps that were generated by Luca Cavalli-Sforza and his colleagues in analyzing European data, first in 1978 and later in 1994. And so what they observed in their maps were, in particular the 1978 paper, is largely based on seeing the PC1 map showing a gradient in this direction. And they interpreted this as evidence for that Neolithic expansion that I talked about early on, of farmers moving from the Near East out into the Northwest. And the kind of rubric that they used for interpreting these maps was, if there's a radiation of circular elliptical lines from a specific area, a population expansion is a possible explanation, and its place of origin must be the center of the radiation. So they saw these radiating lines and thought, oh, this is evidence of the expansion, because it lines up really well with the archaeological data. They also looked at PC2, and they saw these lines here and thought, oh, this is maybe the remnants of Uralic populations that entered from northwestern Asia into Europe. Here they saw this centering on that centered on the location of archaeological evidence for the Proto-Indo-European Kurgan culture. And so they thought that this might reflect that expansion. And here these lines they thought represented perhaps a signature of an expansion of Hellenic populations. So, in one of my studies, we proposed that actually these are more likely due to these general patterns that appear in just any data where you see genetic similarity. So that these are 2 perpendicular gradients. These are these mound-like shapes that show up in PC3 and PC4. These are saddle-like shapes that appear quite common as well. So you actually look at the patterns that they got. These are also from Cavalli-Sforza's work. The patterns they saw for Asia and Africa, and again you see for PC1 and PC2 perpendicular gradients in Asia. For PC1 and PC2 in Africa, you see a gradient, a largely perpendicular gradient with this outlier area. You see this saddle-like shape in PC3 of Asia. It seems to be somewhat in PC4. And then the mound-like shape is present in PC5. These are actually the mathematical forms of what they should like as compared to the simulation, the simulated versions that I showed earlier. Anyway, you see some evidence of these mound-like shapes, but the main point is that these perpendicular gradients appearing in PC1 and PC2 are really a feature of spatial data in general and don't necessarily reflect range expansions. And so there's a disconnect here between the population, or inferences of population processes, and what we're seeing in these PCA descriptions. Okay, so now I want to talk briefly about a couple other features of the data. So some of you might have noticed these 5 Italian individuals that are sitting here and wondered what are they doing. We wondered ourselves, like whether they might be Sicilian or Sardinian, or reflect some insular Italian population. And so to follow that up and actually begin looking more generally at more populations within Europe, we've been looking at, we've actually merged in data sets that exist for Sardinians with this data set. And so you see here that these 5 Italian individuals lie quite close to Sardinian individuals that are sampled from the Human Genome Diversity Project. And you can also see here these black Italian and Tuscan individuals are from the Human Genome Diversity Panel. And an important thing here is that they line up with the Italian individuals from the previous GSK-sponsored study. So that actually reflects that we are able to merge the 2 data sets successfully, which is actually a big challenge because of different biases in the genotyping platforms. You can get sometimes the PCAs just reflecting what center did the genotyping rather than population structure. So that's a kind of a not so great issue when you have in some cases where the data is not high quality, but in this case we're able to merge the data successfully, and we see this. So more generally, the Human Genome Diversity Panel includes a number of isolate populations, whereas I mentioned most of our samples were from Lausanne and London, so they represent European sampled in large cities. So you have this interesting contrast between the 2 data sets of isolated populations versus more urban ones, and so we decided to look at isolated populations more generally, and we also included the sorbs, who are a population isolate located in eastern Germany that have been hardly studied at all genetically, but some collaborators of ours, a group at Leipzig asked if they could collaborate with us to look more carefully at their genetics. So we're going to look at the sorbs from eastern Germany, the French Basque from the Basque region, Sardinians, Orcadians from the Orkney Islands off the northern end of Scotland, and the Adyiegi, who live in the Caucasus Mountains, southern end of the Caucasus near the Black Sea. And you see here in terms of these, how do these populations fit in with their geographic neighbors? So most strikingly, the Basque are actually quite different from their French and Spanish neighbors, whereas Orcadians are not particularly different from other individuals from the UK and Ireland. The Adyiegi are sort of out here with respect to the European sample, but to be honest, we don't have many samples from around the Black Sea, so I can't say whether this is population isolation or that we just don't have enough samples, that this doesn't reflect just a sampling gap and there's actually a continuous range of relationships across here. Interestingly, the sorbs, which they speak a Slavic language, even though they're surrounded by German populations in eastern Germany, they have retained a lot of unique cultural traditions, and their origin have been uncertain. Some have claimed that they may have come from southern Slavic groups. They actually appear on the plot to be exactly where you'd expect geographically, that they look like German and Polish individuals. And so it appears that regardless of their cultural traditions, they're genetically quite similar to their geographic neighbors. So really out of all of this, of these isolate populations that we studied so far, the only ones that are substantially clearly isolated are the Basque and the Sardinians. And this really might just reflect the time scale of differentiation, that the Basque really are thought to be quite an old population and maybe even representing relictual Paleolithic populations. And so we can actually detect that differentiation, whereas some of these more recent isolates, the Orcadians, the sorbs, might either have been isolated too recently, or also have had enough back migration with their neighboring populations that they don't appear genetically distinct. Okay, I'm going to skip this next point because it's technical and not as interesting. And so just to summarize, these large single nucleotide polymorphism studies are allowing us to understand population structure at very fine scales. And I bring this up in this audience particularly because I think that there are many anthropological hypotheses that might help be empowered by being able to study the exact relationships amongst the groups in question, and sort of the microscope that genetics provides to anthropologists has just gotten a lot finer. So when you think about studies like the studies of the Lemba in South Africa that have an oral history of being related to Judaic groups in from the Mideast, and Y chromosome studies were able to show evidence that their oral history was actually correct. Those kinds of studies can now be made much more powerful because of the scale at which we can detect population structure. And so, and then we're still looking at these isolate populations and trying to do a more careful job of understanding the relationships between isolates and their neighboring populations. So in particular, we're trying to estimate the time of divergence between an isolate and its neighbor and get at the timing and magnitude of migration between those populations. Okay, so now I want to switch gears and talk about the role that population structure plays in the adaptive process in humans. And this is work that comes out of collaborating with Jonathan Pritchard's group and is part of analyzing, looking at the Human Genome Diversity Panel. Again, these 52 populations around the world. Before I was just talking about some of these, or you can see the Orcadians, the Sardinians, the Yagi. But now we're looking at all of these 52 populations across the world and looking at their allele frequencies for 650,000 SNPs. And we have analyzed patterns of selection in this data and can put it into a genome browser that you can look at on this page. But I'm going to kind of expand the discussion a little bit here and talk more generally also about some published work on lactase persistence and skin pigmentation, and then bring in some patterns from that data set first. So getting back to Darwin, there was a missing genetic basis in his theory about he did not understand what the genetic basis of evolution was, and we still are just making progress in answering this question, what are the molecular genetic changes that take place when a phenotype evolves under natural selection? So in other words, what is the genetic basis of an adaptive trait? And when a novel environmental pressure begins, you've got three sources of adaptive variation. The first is standing variation. The population could have just had variation within it, that when the selection pressure begins, what was previously just neutral variation, all of a sudden one of those variants has an advantage over the others and begins to spread. Okay. A second one is that the population won't even respond to selection because there's no variation until a mutation enters that has a fitness advantage, and then that begins to spread and respond to the selection pressure. The third is that we have an immigration of an adaptive allele. Okay, so instead of waiting for a mutation, the population has to wait until an allele that is adaptive comes from a neighboring population. And how these three different pathways and how a population responds to selective pressure is going to determine what are the genetic basis of adaptive phenotypes, whether we have for a particular adaptive phenotype many different mutations that explain it or just a single one. So to help make this a little more concrete, the example of lactase persistence is a really interesting one. So lactase persistence has been studied for some time, and human population geneticists have been able to understand the molecular genetic changes that give rise to it in at least a few populations. So there is the lactase gene, which digests lactose, is located here, and within an intron of the MCM6 gene upstream, it's been found that multiple mutations affect how lactase is turned on in adulthood. So this is really interesting. It's not a change in the lactase gene that makes a difference. It's a change in the intron of an upstream gene. Now it's thought that that intron matters because that there's that those sequences have a regulatory role over lactase. And what's even more interesting is that the mutations are very closely spaced together. So here we have this CT mutation explains lactase persistence in Europeans. So individuals who have the T allele express lactase as adults, therefore they're able to digest milk as adults, and this presumably has given them a fitness advantage based on the fact that you can see really strong signatures of natural selection around that mutation in European populations. Now, Sarah Tishkoff and her colleagues have studied lactase persistence in other African populations, and in particular have picked up at least two different mutations, additional mutations that that can give rise to lactase persistence, and in Kenyans and in Tanzanians. And so we have this, interesting feature that the same adaptive phenotype is being explained by unique mutations in different regions of the world. Okay. And now let's look at skin pigmentation. With skin pigmentation, first off, this is just looking at a list of genes that show the strongest signatures of positive selection. And amongst those, you have morphological traits, such as these genes that are involved in skin pigmentation and hair development that are on that list. And so these are some of the most differentiated genes between any human populations. And it brings up the interesting point that what we see as surface indicators of race, so skin, hair, and eye color, are some of the most unique regions of the genome. If we make a list of the most extremely differentiated genes, up pop these morphological traits. And then, so if you look at this more carefully and you take skin pigmentation as one of as a trait, the genetic basis of skin pigmentation has been is increasingly being better and better understood, so that now we have at least this many loci here. It's about, I'm counting from here, 10 or 11. So, but these are the main well-studied ones that are have strong support for them being involved in skin pigmentation. And if we ask what's the distribution of FST values across the genome, so SNP by SNP, we ask how different are the allele frequencies between different populations, Africa and Europe. Here we have a full distribution, and then we plot where the SNPs that are involved in each of these skin pigmentation genes sits. We see that some of the skin pigmentation SNPs are some of the most extremely differentiated ones in the whole genome. So this is reiterating that same point. And but further, if we plot maps of some of these genetic distributions of these SNPs, we see these really striking patterns. So for instance, KIT-LG, one of the mutations, has this pattern where it's distributed outside of Africa in really high frequencies and is almost absent within sub-Saharan Africa. SLC24A5 has this pattern where it's seen in Western Eurasia, but not in Eastern Eurasia. MC1R has this pattern where it's seen in East Asia and the Americas, but not in Western Eurasia, Africa, or Oceania. Now these clusters, these appear, these line up roughly with genetic clusters that have been inferred previously from large numbers of neutral markers. So if you know the study by Noah Rosenberg and his colleagues, they saw major geographic clusters around the world, and these match up with the Western Eurasian cluster that they saw, and the East Asia cluster, and the Americas cluster is thought to be closely related. So the point here is that, probably more importantly, is that European populations and East Asian populations and the American populations essentially have lighter skin color for different reasons. They use different mutations to get light skin color. So you have to ask, why is there not just any one of these mutations that gives lighter skin color spread across all of the northern latitudes that require lighter skin color? That's the real interesting puzzle here. And it basically seems that the history of gene flow in humans has been affecting this geographic distribution. That presumably as humans colonized out of Africa and began moving around the world, you had mutations that arose in this population that spread out through here to give lighter skin color, but that didn't spread to East Asia. So it appears that population structure has been somehow limiting the spread of adaptive mutations and making the genetic basis of adaptive phenotypes more complex. It's not that there's one mutation or a small number that can explain it. You actually have several mutations arising in different locations to lead to the genetic differentiation. So that's what the pattern that we saw in lactase. Dairy culture is very recent, so you have different lactase persistence mutations arising in different parts of the world and giving a response to the phenotype. And the same thing going on here with skin pigmentation. So some adaptive alleles appear to be more geographically restricted than the selective pressures that favor them. And that's likely due because population structure is slowing their spread. And that's probably especially true for these examples like dairy culture, where if the selective pressure is very recent, there's been little time for these mutations to spread more broadly across the world. And this leads to this complexity that you might have different genetic mechanisms that may explain different phenotypes in different parts of the world. And this could be especially true for traits that have a high effective mutation rate. So if the way that you can get a phenotype, an adaptive phenotype, is only by a single mutation at a single base pair, it might be that the mutation arises once and it will have time to spread across the whole population before a second mutation arises. But if it's something like lactase, where there's this promoter region where a number of different mutations will allow it to, the lactase gene to be expressed into adulthood, you have a higher effective mutation rate, and it's possible then for multiple adaptive haplotypes to arise and spread in different parts of the world. So you have this interesting interaction of population structure, mutation rates, and selection that give rise to the genetic basis of adaptive traits. And we really need more studies of adaptive phenotypes and what their genetic basis is before we can really evaluate these hypotheses. One of the real challenges in this, in making further progress in this area, is that we have strong biases in what we can actually study. So, with this analogy of looking for your keys, you drop your keys in a parking lot, the only place you can look is under the lamppost, because where it's too dark you can't tell it's there. So the same thing is going on in these studies generally, in that if you think about, you can classify selection into what some people call hard sweep, which is that we imagine that before selection, these are just the chromosomes that exist in a population. After selection, we have an advantageous mutation that's arisen and has spread. You see that's fully fixed in the population. And there'll be these regions that are attached to it that have this genetic hitchhiking signature that are that basically when a mutation arises, it lands on a particular background. And if the mutation increases in frequency really quickly, then that background will be carried with it. And so you'll have all of these mutations that have the exact same genetic background. And this kind of signature is very easy for us to pick up. This is the kind of signature that you see at lactase, and that makes it one of the most well-studied examples of selection in humans from a genetic level. But if adaptation looks more like this, where before selection even begins, you have some variation that is going to become adaptive. Okay, this is sort of representing the standing variation that I talked about, that you already have variants in the population that will help you respond. And then moreover, it's distributed across many different loci. Then, first off, when you have the spread of a standing variant, so this one, it increases from a frequency of 1 to 3, you don't get the same hitchhiking signature, so that's not present. And then also, even where you do have a new mutation arising and increasing in frequency, the frequency increase doesn't need to be as large, because other mutations are going to be pushing the population or individuals towards having the adaptive phenotype. And so instead of having, you can essentially distribute the adaptive response across many different genes so that none of them show a strong signature. And so we'll miss out on all of these. It's very difficult, at least with the techniques we have right now, and potentially with any technique you could imagine, to be able to understand these, or to detect when this is happening. So, we've got this challenge to studying these polygenic adaptations and where you have standing variation. And this is important because it's the kind of the alternative, right? So if standing variation is important, well, it might be important, all right? It's actually what it's suggesting. Because a lot of human adaptation has been to very recent environmental change, there's been little time for novel mutations to occur. And there's also been little time for the alleles to spread between populations. So a lot of the responses that we see, you might think could be due to standing variation rather than novel mutations, since it might seem unlikely that we've had enough mutations in such a short amount of time. And we know that, remember I talked about these 3 pathways, because of population structure, it's unlikely necessarily that many of them have spread between very distantly spaced populations. So an interesting part of that follows from that is that most standing variation in humans has been shared among populations because of our recent common ancestry in Africa. So a lot of the standing neutral variation that we have is completely shared across populations. So if that's the case, then the genetic basis of adaptive traits will look very different from what we saw at skin pigmentation. You would have variants that are standing in the population, they're shared across the globe. When a selective pressure like skin pigmentation arises, or a pressure for in northern latitudes for lighter skin pigmentation, you would have lots of genes changing in making small frequency shifts, and you wouldn't have these distinct partitionings of adaptive alleles into geographic populations. So the genetic basis of adaptive traits would be more shared among populations, and in that case, these lactase and skin pigmentation examples that are the best examples we have that are well studied might be the exception, not the rule. So we're really trying to bring up an interesting area which human population geneticists are puzzling about right now, because we're right at this exciting time where we're getting lots of new methods for making better associations between genotypes and phenotypes. That's why we understand the mutations that underlie lactase and underlie skin pigmentation. But we just have these few examples. They're showing these interesting patterns, but we also know that there's a whole set of traits that we haven't studied and that we have biases in what we can learn about them. So it's going to be really interesting to see what comes out of this. And so we just need more well-studied examples of adaptations part. And then also there are very few theoretical models that try to integrate these competing processes of when you have a novel selective pressure and you have mutations arising potentially in different parts of the habitat with migration slowing their spread. I think that's going to be an interesting area for future work, and something I'm interested in doing. So with that, I just want to acknowledge a lot of the people who are involved in a lot of these large studies. So the Popper's European Diversity Project involved Matt Nelson, Carlos Bustamante's group, and Bergman's group, Matthew Stevens, who's working with at Chicago, and many other lab members. The population isolates work has been done with Krishna Varama here at UCLA. He's in the Center for Society and Genetics and in my lab. Collaborators at Leipzig for studying the sorbs. Part of Michael Stombol's group. The Human Genome Diversity Panel selection analysis was mainly run with Jonathan Pritchard's group, with grad students Joe Pickrell and now Professor at UC Davis, Graham Coop, contributing a large part, and of course thanking funding. So, thanks.